Hello and welcome to Nikolai's genetics lessons, and today's question is: diagnosis of chromosomal aneuploidy of unborn children is normally done by combination of amniocentesis, cell culture, and you have to choose one of these techniques. Here on this picture you see how amniocentesis is done. Here is a fetus which is in amniotic sac and surrounded by amniotic fluid. And in this amniotic fluid the cells we can find which this fetus is shedding, and by taking a sample of this liquid we also take samples of the cells of the fetus. And in order to find the position of the fetus we also using ultrasound in order to put syringe in the correct place. For example, we cannot put it here, but this is going to be the best place according to this picture. And next step would be to culture cells. And now let's talk about these procedures just very briefly. By doing enzyme assay, we can find which enzymes are present and which are missing. So usually enzyme is specified by one gene. Sometimes enzyme can be specified by a number of genes if it is a complex enzyme, but this technique is good in order to find diseases which are caused by one gene. But we are here looking for chromosomal aneuploidy. Aneuploidy means that during meiosis, when chromosomal pairs would be pulled to the different poles of the cells, instead of being pulled to the different sides, for example, this one chromosome would be pulled to this side. That means that, for example, when gametes would be formed, this gamete is going to miss this chromosome, but another gamete is going to have extra chromosome number 2 instead of just having one. In both scenarios, when one chromosome is missing or present in extra copy, we call this aneuploidy. So will restriction fragment length polymorphism analysis will help us to detect extra copy or missing copy of chromosome. Imagine that this is double-stranded DNA, and let's say here is going to be a restriction enzyme recognition sequence. And if this sequence would mutate, for example, instead of G here, it's going to be, for example, A, adenine here. And now restriction enzyme is not going to be able to attach to this recognition site and is not going to be able to cut it. So with this technique, restriction fragment length polymorphism, we can obtain, if we have a long enough DNA sequence, restriction fragment length polymorphism of the chromosome. For example, in one digestion we can get following chromosome fragments, and with the same enzyme, restriction enzyme, if we also cut the same DNA fragment from another person, we can get different picture. And here, for example, this person is going to have a mutation. If this mutation would be strongly associated with a gene, for example, here is a gene, and because it is so close to this gene, it would be tend to inherit together. And if we know that this mutation is very closely associated with certain disorder, genetic disorder, we can predict that such restriction length polymorphism of this fragment of the DNA would cause certain genetic disorder in person. This good technique in order to find only one disease, which is usually caused by substitution of one nucleotide with another, so single point polymorphism or mutation in single point, maybe 2 nucleotides, maybe 3, but this technique is not going to tell us if we have chromosomal aneuploidy. Next, pedigree analysis is also a very powerful technique, for example, to find probability of the child to inherit certain genetic disorder if we know the history of this disorder in a family. But again, it's not going to tell us anything about chromosomal aneuploidy. Why? Because people with extra chromosome or missing chromosome have very severe health issues, and such people usually cannot have children of their own, so this technique is not going to help us in search of the chromosomal aneuploidy. Let me skip D for now. Somatic cell fusion, it's also very powerful technique. For example, we can use this technique in order to find positions of the genes on certain chromosomes, and we hybridize human cells with mice cells. But again, we are not using this technique in order to find chromosomal aneuploidy. Instead, we are using karyotyping. Only karyotyping is going to show us the whole picture of the chromosomes in a cell. Karyotyping is going to help us to find, for example, extra chromosome 21. This is Down syndrome. Sometimes we can find missing X chromosome or extra X chromosome. We also can find circular chromosomes, and we also can find if two non-homologous chromosomes exchanged arms. So with karyotyping, we can find major changes on chromosomal level, when the whole arms would be exchanged or missing parts of the chromosomes, which can involve hundreds of genes. Because chromosomes, many of them look alike, we're using special dyes which reveal certain pattern of these chromosomes, and we can arrange them in homologous pairs. Usually amniocentesis performed on the women who is over 40 years old, because with the age, probability of different genetic disorders such as trisomy 21 greatly increase. So as you see, our choice is answer D. And this is all for today. Subscribe and see you in the next video. Goodbye.